chromatography is what we're looking at. Now, big picture for me. What is chromatography? What's the whole point of chromatography? Why in the world do we study this idea? What are we trying to do?
Separate things.
We're trying to what?
Separate things.
Trying to separate things. What things? Proteins. And so we're trying to isolate a protein. Okay, so what I have is a column. Okay, it is as small as a little tiny pipette. Okay, or I've used ones that are this big, depending upon what you're doing and how much you're trying to separate. All right. And so, what is the separation? Where is the power of separation in this?
So the size of the proteins?
Hmm?
The size of the proteins?
Okay, it could be the size of the protein. All right. What we're going to do is we're going to identify one physical characteristic of the proteins and separate based upon that. So this one physical characteristic could be size, and we separate that way. One physical characteristic could be charge, and we separate that way. One could be what it binds to, its hydrophobicity, how polar or how nonpolar it is. There are lots of different physical properties I can separate proteins by. Okay? But where do I actually have the interaction that can separate these physical properties? Where is it happening in this model? It's on the matrix. All of that material, you see those little tiny spheres all over here? Those spheres will be coated, and they're very tiny because the smaller you have, the more surface area you have. And we use polymers of sugars, really, and then we coat the outside with certain things, or we do have those pieces of sand that might have holes in the interior, depending upon what we're doing. All right, we're going to coat them. And so some of the proteins are going to be attracted to that, what's called the solid phase, okay? And they will stick to it. Some doesn't stick to it. And so when you put your material, that mixture of proteins on the top, what's going to happen is everything's going to kind of be there and stick to it, okay? And because it's actually touching. But how do I actually know if it's actually making an interaction that wants to stick to the beads or is just sitting there because it's there? What do you have to do to make things move through the column? Yeah, you have to pour solution or buffer on the top. And we use a buffer solution to keep the pH correct because remember, you change the pH, you can change what charge is on the protein or you can change its fold. So we have to keep the solution at a certain pH. And I'm going to run buffer on the top, put it on the top, and I'm going to open up the bottom and it's going to flow through. If proteins don't interact anything with the matrix or those little spheres, what is that flowing buffer going to do to those proteins? It'll push them through, right? What we talk about is washing the column. And if you think about washing, we're going to clean it. What we're going to do is we're going to wash through the material that doesn't stick. Okay, so running buffer is something that we talk about. That's the solution we put on top. It runs through the column. We're going to wash the column off of anything that isn't doing the interaction that we're particularly interested in. But, oh, good night. Now I've washed the column. Some of the proteins will run off, right? But some of the proteins are stuck. Is that where I want my protein to be, stuck on the column? No. So what in the world do I have to do? Okay, the next term that we talk about is eluting. Okay, we're going to elute. Now what happens is if you just use your running buffer, all right, you wash off everything that doesn't stick. But if you want to get the stuff that does stick, you have to change buffers into a new buffer called the elution buffer. That means something in the buffering system is going to be added into the solution that will displace the things that are interacting with a solid matrix. Okay, it's also called the mobile phase. The solutions that run through are the mobile phase, and the matrix is called the solid phase. And depending upon what you add as the elution buffer, you can elute things differentially. Okay, you can either get all of them off at one time, or you can sequentially knock some of them off, and then knock more of them off, and then knock more of them off. It all depends upon what you need and how much separation power you need. All right. And now, so the other part of this is taking the fractions. You can do wonderful elutions. You wash it, elute it, but if you collect it all in one big beaker at the end, even though they come off at different times, you'll have no separation because they'll all go into one big pot. You have to take fractions. The more fractions you take, the better resolution you can have. Okay? And so, this is the general idea. So, I usually wash the column with running buffer before I put anything on, just to make sure everything's moved out. I load with my mixture of things I want to separate. I wash with running buffer to wash off everything that won't stick. And then I elute using a new buffering system. And then I collect fractions, and then I usually wash the column to make sure it's all clean before I start again. These columns can be reused, and are reused because the matrix are sometimes very expensive. Okay, the other thing, one more thing about this. This is a gravity fed, so you've got a column and you just pour it on top and you allow the gravity to flow water through, or the buffering system. We can also hook them up to pressured systems, all the way up to high pressured liquid chromatography, HPLC. We have some in the lab that we use. The higher the pressure, the smaller the surface area of the matrix, the more resolution we can have, the better separation we can have. But most of the time, gravity fed is good enough for us. But sometimes you need something that has more pressure, and we have full systems to do that. The first kind of separation is called gel filtration chromatography, or size exclusion. All right, so size exclusion tells you what we're going to do. We're going to separate by size. Okay. Now, we're going to separate by size in a couple techniques. In this case, largest molecules come out first, smallest molecules come out last. Okay, that's not always the order, but in this case, the order is big out first, small out last. All right. The matrix, or the little beads, will be hollow inside and have pores. Okay. Now, one thing you can do is you can go to the catalogs and buy different kinds of beads that have different sized holes that will separate fractions of molecular weights. And usually we talk about molecular weights, although that is not truly exclusive. Okay. What it's going to do is going to separate, yes, by size. Most of the time it's this. The smaller molecular weight is the smaller size. The larger the molecular weight, the larger size. This is true if and only if the molecules are globular. Because what's going to happen is your protein is going to be in solution. Okay, it's got a certain shape. And it falls by inside that running buffer. And so it's moving in that volume of liquid. What will happen is that protein will tumble as it falls. The size is the volume of the tumbling. Okay? And so if your protein is globular already, as it tumbles, it doesn't change size very much. If by chance you had a protein that is long in one and short in another, then as it tumbles, it will take up the radius of the longest end. And it can actually look deceptively large because it takes up a large volume as it tumbles. Okay? But in general, it's usually smaller size to larger size because most proteins are globular. You can have some oddball things happen if the protein isn't truly globular. Okay? And so, if you take a look at the animation at the top, you can see the colored material as they fall through. You can see that you get a fraction of different sized proteins. Now what's important is in terms of resolution that there should be a band that is clear between each one of those colors, okay, if you want real good separation. And here's the problem. Your protein mixture starts off in a buffer solution that looks like water, clear, and it goes through all of this, and all of your fractions are clear. You don't know if it's actually separated because you don't see color until you do analysis. And so you have to analyze every fraction to see if there's protein there or not. Okay? And so what you hope to see is fractions that have no protein, and then fractions that have a protein, and what it does is it will produce a peak. If you plot the fractions versus concentration, you have a peak of your first protein and come all the way down and show no protein. Then you'd have a second peak with your second protein, and then no protein, and then you have a third peak, and you would have separated the three proteins very well. The problem is when you actually really do it, you get this peak that comes up and then starts going down, and then all of a sudden a peak comes up, but they're overlapping. That meant you didn't get good separation. And so what does that mean? You try again with something else. Okay? And so you place your sample on, and in this case, the running buffer and the elution buffer are exactly the same. And so it's just volume of buffer going through that will push everything through. We're not sticking like we're going to do with the others. We're just falling through. Now, I'll show you this animation. Again, this will be linked on the Moodle site. And at the molecular level, there is this music going on all the time. All right. Here we have different sized proteins. The larger ones don't fit inside the holes, so they just go around. That's why they can go faster. Now here, you can see up here, there's nothing coming off the column yet, but those red ones are going to come off the column, and you can see, if I take fractions, I can see concentration. It comes back down, there's no protein, and then here's the next protein. And as I add more and more buffer, or more fractions that are taken, you can see the little ones get held up. And it takes more buffer for them to go. They actually fall through a larger volume because they not just go around, they fall inside like a little thing, what is it? Pinball machine. You cannot hit, and it takes them a lot longer to come off. And so you can see the three proteins are isolated by size. Yes?
So why did the green fall through it so much faster?
Because in there, the holes are small, little, and tiny. And so if the green ones get in there, they may only get through the passageways that are rather large, whereas those blue ones got into the passageways that got into the smaller ones and the smaller ones. And so it is honeycombed with different sizes. And so the green one didn't have to fall through as much, but the blue one had to fall through every single hole that it found. And it's the design of those beads, what those holes are. Now, for every type of gel filtration, you get and you purchase, I'm going to separate between this molecular weight and this molecular weight. And so, and then you can buy different ones that will isolate. And so that means at the cutoffs, anything smaller than X will fall at the same rate. Okay, from 0 to, let's say, 10,000, all of them fall at the same rate. And then at the upper limit, it might say 50,000 molecular weight units. Anything that is 50,000 or larger, 100,000, they would all fall at the same rate. The only separation you get is between 10,000 and 50,000. That's where you actually get separation. And so, but you can buy all sorts of ranges, and you buy the one most appropriate for the one that you're looking for. And so if yours, you can actually separate a whole lot of proteins that are very, very large by getting a cutoff that is pretty low, because all those big proteins that you don't care about because your protein is much smaller come out at one time, and yours is way down in a different range. And so it's a pretty powerful way that you can do this. Okay? In this case, you can see things might have to fall through all of the holes while the big ones fall around. And so, and essentially the big ones fall through a smaller volume of space. This technique is as often used to desalt a protein as dialysis, because salts are very, very tiny, hundreds of molecular weight units, whereas the proteins are in the tens of thousands most of the time. And so running through a column like this can also be as effective to desalt as the dialysis that we saw. All right. The next, ion exchange chromatography. What does ion exchange make you think about? Charges. Okay. We're going to separate by charge. All right. So you see that the bead is full of positively charged ions. So that means your mixture of proteins, everything that is positive will fall right through. Anything that is negative will stick. Now, if you take a look at these, some of these have 1, some of them have 2, some of them have 3 charges. Now, what are you seeing falling off right here? Only the charge that is 1. Now, this line represents increasing salt concentration. Okay, and it's a gradient, starting from 0% salt to a certain amount of salt. And as the salt concentration goes up, what do you see falling off? Higher and higher charges. And so if you put a little tiny bit of salt into the elution buffer, only proteins that have a small amount of charge will fall off. Anything that has a large charge will still be able to attract to the bead and hold on in the challenge of extra salt, because the salt is displacing these charges. Okay? I then add a little bit more salt, and I get those proteins that have a little bit more negative charge, leaving the ones that have lots of charge. And then if I add even more salt, I can get the largest charges off. And so this can be a very powerful technique to separate not only positives away from negatives, but even the negatives can be differentially separated by using increasing concentrations of salt in the buffer to elute there. What else could I have added to knock off the proteins? Instead of putting salt in the elution buffer, I could put 2 other things. What? I heard something?
That's a potassium.
Now, a potassium would be just a different salt.
Yeah.
Different salt, but yes, we could use that. Why I use sodium is sodium's cheaper than potassium or magnesium, but yes, you could add either acid or base. Why would that be able to elute something off? What did you learn about adding acids and bases? You actually, because a pKa can either protonate or deprotonate, changing their charge. And so you can either protonate or deprotonate the protein, or protonate or deprotonate the actual bead itself, and you can do that. So acids and bases can be used. We tend not to use those if we don't have to, because adding too much acid and adding too much base, yes, can change the charge, but also could facilitate rapid degradation of proteins. So sometimes you use it, sometimes you don't, depending upon how tolerant your protein is to changes in pH. Most of the time salt's used, but pH can be used also. All right. Also, it is important on this in your running buffer to get the right pH for the running buffer so that your protein is in the right protonated state or deprotonated state so that you can have a good interaction. The other thing, the charged bead can be very, very charged or slightly charged. And so, depending upon if it's a really strong charge or a very weak charge, will allow you to have a little bit more control about how separations occur. And you buy different kinds of beads that'll talk about a strong ion exchange bead or a weak ion exchange bead, meaning a strong, basically a strong acids base, a strong charge, a weaker one. And so the initial separation is between the two charges, positive or negative, and it depends upon which bead you use, which one sticks. When you're separating, you always want your protein of interest to bind. Okay? You want it to bind, to concentrate, because everything that gets washed through gets diluted. Diluting your protein also helps degradation, and you don't want to do that if you're trying to isolate it. And then you want to elute it selectively. Okay? All right. The protein of interest can be released by increasing the salt concentrations. Low net charge proteins will elute first, followed by high. And what we're doing is by overwhelming the solution with, let's say, sodium positive ions, it just, by mass action, the bead will interact with that sodium charge because there's just so many more and displace. You can also use sodium chloride to displace things that are negative. It's just the chloride ion is what's going to displace. And you can buy all sorts of beads, some that have a partial charge, like a carboxy group. That's considered a weak ion exchange. Or you can get ones that are positively charged all the time, a strong ion exchange. And so you look these up and you can buy a whole host of these from different suppliers. Affinity chromatography. Here's a very powerful way in which we can separate. Some proteins bind small molecules or entities very tightly. It's part of their function. And so let's say my protein binds to, let's say, ATP. It binds to it very tightly. I can use that characteristic to actually isolate my protein. Okay, but it needs to be binding that is non-covalent. It needs to be tight for this to work. Okay, and your protein needs to be able to do it, and it needs to be unique enough that it doesn't just pull everything out of the solution. What you do is once you have that unique protein, that unique entity, whatever it is, it could be a small molecule, could be anything, you place it onto the bead, okay, and you link it to the bead. And so now the matrix has that small molecule that your protein of interest will bind. You put the mixture on top, you wash it. Most other proteins shouldn't bind to it at all. If you wash it well enough, the only thing that's stuck onto that bead is the protein that you're interested in. Okay? And then you can elute it. Okay? So here is an example. We have three proteins. All three proteins bind to things, but each protein binds to something separate and different. It doesn't matter what that entity is. But what we're going to do is we're going to isolate one of those proteins. And we have taken the matrix and we've connected that small molecule. Okay? As I put the whole matrix, the whole mixture through and wash it, the only thing that sticks is the one that I want. Everything else gets put through. Now, take a look at the eluting buffer. What I do is I put a very high concentration of that small molecule in solution. What happens is the protein will fall off. Well, why in the world does it fall off? Here's something about biological systems. Okay? When something binds, all right, there is always a dissociation constant. In biology, things are moving and happening. What happens is, here's the entity, here's my protein, I will bind to it, and I say, and it's a tight binding. Okay, that's good. But in biology, I never bind and I'm stuck. What happens is, I will bind and let go for a moment, and then, depending upon what's there, I might bind right back. If there's nothing else to bind to, and it's always a dynamic bind, let go, and there's a dissociation constant, a rate at which that happens. And so when I put my elution buffer here that has that small molecule, what I do is I put the small molecule in the solution at such a high concentration that when this does open up, what happens is there are so many other molecules that are available, it may bind to the one that is free, not the one that is linked. So it's bound to the one that's free, but that now it's bound, but it's free falling. And it falls out the column. At the end, do I have purified protein A, as they say in this test tube? No. You have the protein plus that small molecule. What could you do to get a pure protein? You can either do size exclusion, because that small molecule is much smaller than your protein, or two, you can do dialysis. And so you have one protein, but you have the other stuff in it, you will have to do an extra step. And all of these, even like your protein that was on the affinity, on the ion exchange, you put salt in there. When you get your pure protein, you've actually got salt. You'll have to desalt it using gel filtration or dialysis. So there's always another step. And so here is a picture of that animation showing the mixture on top. You wash it off. The protein of interest becomes bound, and then the elution solution or the elution buffer is placed on top, and then your protein comes off. All right. Sometimes the protein that you're looking for doesn't have an affinity for anything. But wait, we can add something. All right. Now, if you start from a protein from tissue, if you take the brain and you chop it up, you're not going to be able to do this. But if you do it from a cloning perspective, you've isolated the DNA of the protein of interest that you want, then what you can do is add something to the end. All right, just for purification. And a lot of times what we do is we add a his tag. His tag is this. It is a small linear chain of histidines to the end of the protein of interest. It also has a place where you can cleave it so that we can selectively get rid of it when we want to. But adding that series of histidines to the end of your protein will allow us to do a tagged purification or a modified affinity chromatography. All right, let me show you the animation. Okay, that's a minusole to chemists. And so here we have created a tag. And why his tag? Well, one company developed this technique and sells it, and a lot of people started to adopt it. There are all sorts of different purification tags now that we have, depending upon what company that you use. And there are some where you use magnets when you put the tag on, and so you can use a high-powered magnet, flush everything through, all your protein gets stuck to the magnet, and then the purification, take away the magnet, it'll fall through. That's kind of cool. But there's all sorts of different techniques that we can. And it just depends upon which one you want to use. But it is coupled to when you do a genetic or a cloning technique where you're going to make the protein that way. Sometimes even in the studies they don't remove the tags. They continue to use the protein. The tags may or may not interfere with the experiment. If it doesn't interfere and you're lazy, you keep the tag on. But most of the tags have a portion that can be cleaved selectively so that you can get your protein pure away from the tag. All right. The chromatography techniques. Here's the deal. I don't know of one purification scheme that gets your protein pure using one single step. Okay? Most of the time, it's multiple steps to get a pure protein. How many steps it takes depends upon the complexity of the mixture you start with and the competing things. Because you might be able to purify something that you purify your protein of interest that is very, very negatively charged. But here's the problem. In an entire cell extract, there are going to be proteins that are very, very negatively charged also. And so most likely you're going to get a mixture of very negatively charged proteins in your mix. Well, they're all very negatively charged, but your protein might be very, very tiny and all the other proteins may be very, very large. So then you do a next step of size exclusion, and you get rid of all the big ones and you get to yours. Usually it takes a combination of 2 to 3 to 4 different separation techniques to get to a purified protein, a single protein by which you can use to do your studies. Okay? And so, but they're used in conjunction. And which ones you used, it depends upon, again, you hope someone's isolated your protein before and you just find out their protocol, they figured it out. If not, you do a lot of work to figure out how to isolate your own protein. All right? These techniques, salting out, dialysis, chromatography, are all used for large amounts of active protein. So you're getting enough to study. Now, what's large amounts? You may start off with a kilogram of material, and after all of the purification, you may end up with a milligram of your protein. That might last you a couple months worth of testing. Okay? The isolation is not that, you don't get a lot, okay, most of the time. In terms of most proteins, you just don't get a lot in this process. So quite often, if you are studying a protein, you're doing a lot of purifications all the time to make sure that you have the source. Very rarely do you buy something directly, even if you buy it from a company. It's not at a purity level that you really need it to be. And then you put all of your work based upon their work, and that may not be exactly what you want to do. So you're usually isolating things. Electrophoresis is an analysis. It's going to be a separation, but it's going to be analysis. It is high resolution, much higher than anything that we've seen up to this point in time. That means I can separate very small differences. Okay. It is also going to be very tiny amounts, and by the end of the process, the protein is inactive. Okay? And it's not functional anymore. Now, the protein is there. We can detect what's happening, but I'm not going to, from that point on, study the protein's function. This is usually the end result of doing an experiment and then using electrophoresis to see what happened, because after this, the protein's dead. Okay? What we're going to do is we're going to purify proteins using electricity to move them through a matrix to cause separation. All right? And we're going to use that electric field so that they move, and we're going to try to move negative ions towards the positive electrode. Now, what do you know about proteins themselves? If I put a protein in a solution, what charge might it be?
Does that depend on the solution?
It really depends upon the solution and the protein. A protein can be highly positive, a protein can be highly negative, a protein can be neutral. But I'm going to use electrophoresis to move all the proteins eventually in one direction. Okay? That's the traditional electrophoresis that you can think about. But I'm going to use a technique of electrophoresis to actually work on a mixture of proteins that have all sorts of different charges, and we're going to separate in a different way than maybe you've seen it described before. But we're going to use electric fields to move, and we're going to use the matrix to separate. Now there's a difference. Okay? Movement doesn't cause separation. It is the interaction with the matrix. In this case, what we're going to do is we're going to use either agarose or we're going to use acrylamide. Most proteins use acrylamide. And what it's going to make is probably, how many people have actually interacted with an electrophoresis apparatus and done some separations? Okay, so the matrix, when you do, it kind of feels like jello, right? And so that jello is what's actually going to cause the separation. The jello itself has different sized holes. Okay? The electric field just makes the proteins move, and if there was no matrix, they would just move and there would be no separation. But as it moves through that matrix, that jello, the jello is what's causing the separation, because things can't all move through the jello at the same rate. We're going to do 3 types of electrophoresis. 1, isoelectric focusing, the gel electrophoresis that we think about, and then we're going to couple the two together. Okay? Isoelectric focusing. All right, here we have a bunch of proteins. We usually start it kind of in the middle-ish. And the proteins all have different charge. Now the matrix is a jello, and what we're going to do in this jello is not create sized holes. The sized holes are going to be big enough that that's not going to separate anything. But what we're going to do is we're going to pour this jello, and this end of the jello has a buffer system that is very basic. And I create a gradient of pHs in the jello all the way to the end that's very acidic. So I go from basic to acidic. Okay? I then put my proteins into the jello, and I turn on the electrodes. Any positive proteins go to the negative electrode. Any negative proteins go to, negative proteins go to the positive electrode, positive proteins go to the negative electrode. And they will migrate. Okay? But as they migrate, they move into different pH ranges. And remember, as it goes into different pH ranges, each R group and each place that can have a pKa starts to protonate and deprotonate, protonate and deprotonate, until you reach a point at which the protein will not move anymore. Why will it not move? It becomes neutral. What is that state called? I've reached the protein's what? P what?
Yeah.
Pi. There is a certain pH at which a protein will have as many positives as negatives, and it becomes neutral. It is not that it's not charged. All of these proteins will be charged, but I have as many positives as negatives, so overall it's neutral. And so I can separate all sorts of different proteins by their PIs, which has nothing to do about size. Okay? But it really has to do with what the R groups are. Okay? This is how we usually calculate a PI for a protein. We don't actually go in and do a mathematical calculation, although you can. What we do is, this protein is at a pH of X. Well, its PI is that pH. And so we functionally identify proteins. And we have all sorts of proteins. If you don't have a lot of proteins, you can get wonderful separation. If you have a very complex, it's separated, but it's messy. Okay, because you get so many in a short distance. And we can cut those out and we can isolate those, but we can separate based upon charge. Here's an example. A lot of times they do it in capillary. The pH gradient goes from 9 to 3. I put them on, turn on the electrophoretic, the electricity, and they will separate, creating bands. And so at the end you get separation. Can be done more rare, this kind of separation. It's not as useful as the next. Okay? It will create and identify the isoelectric point. What we really think about more often is what's called SDS page. Okay? And so I will talk about what the SDS page means next time and go through that process. What slide did I get to?